Now, in the second part of the neuronal transmission series, we are going to continue our discussion of electrical transmission along a neuron and from one neuron to another. Now, in our previous discussion, we had discussed about the mechanism by which an action potential is generated on the neuronal membrane and the mechanism by which it is propagated along the axon from the cell body to the nerve terminus. This chain of action potential, which is shown here along a single neuron, is known as the spike train. The next question which arises is, how is the intensity of a stimulus reflected in the nature of the action potential? So, all stimulus are not going to be of equal intensity. So, how are stimuluses which are of different intensities reflected in the nature of the action potential? Now, as is shown in this figure, if the stimulus is below the threshold, it does not stimulate an action potential. Only when the stimulus is above a certain threshold, an action potential is stimulated. And when one further increases the intensity of the stimulus, the frequency of action potential is increased compared to that which is obtained with a less stronger stimulus. Therefore, the stimulus intensity is being coded by the number of impulses per second rather than by the increase in amplitude of individual action potentials. Now, this is illustrated in the experimental data which is shown here. Now, if the stimulus is below the threshold, no action potential is observed. If the stimulus is above a certain threshold, we observe an action potential. When we further increase the intensity of the stimulus, more frequent action potentials are observed here. A further increase in stimulus leads to rapid appearance of action potentials and increase in frequency of appearance of action potential. Therefore, this brings us to the universal property of a nervous system, which is the pulse, called the pulse frequency modulation, according to which the nervous system encodes information regarding the strength of a stimulus, not in terms of the changes in amplitude of a particular action potential, but rather in the change in the frequency of the action potential. Now, this brings us to the concept of graded action potential. Now, suppose we are stimulating a neuron at a particular place, and this depolarization is spreading in the adjacent regions. However, this is a local effect, as is seen here, which is very similar to that observed in graded potential. What happens in graded potential is that the stimulus which is received by the dendrites of a neuron from the previous neuron, in this region of the cell body, it acts over short distances, and it is the summation of multiple stimuli which reaches these dendrites. When this summation reaches a certain threshold, they sum together and determine whether an action potential will be generated. So, if the voltage depolarization is triggered, then the graded potential has reached that certain threshold. After that, the action potentials are generated which travels over long distances and with its amplitude remaining the same with no decay which occurs with distance. So these are the action potentials operate over long distances, whereas the graded potentials are weaker depolarizations which occur over short distances. The summation of the graded potential when it crosses a certain threshold gives rise to the action potential which travels down the axon. Now, the next question which comes to mind is whether the effect of a presynaptic neuron on a postsynaptic neuron, whether this effect will always be excitatory or can it be also inhibitory. Now, the extent of the changes which are brought about by the presynaptic neuron on the postsynaptic neuron depends upon the amount of neurotransmitter that is released by the presynaptic neuron into the synaptic space, and also the duration for which this neurotransmitter reside in the synaptic space. Not only that, the nature of the neurotransmitter which is secreted determine whether the postsynaptic neuron will be stimulated or inhibited. If the postsynaptic neuron is excited or stimulated, it is called the excitatory postsynaptic potential, whereas when it is inhibited, it is known as the inhibitory postsynaptic potential. Now let us look more closely into the excitatory synaptic potential or EPSP. Now during the generation of such excitatory potential, suppose first a chemically gated ion channel opens in the postsynaptic neuron. So the ligand binds to this ion channel, the ion channel opens, and this allows the movement of both sodium and potassium ions across the membrane in opposite direction. This results in a slight depolarization of the postsynaptic membrane, although it does not reach the threshold which is necessary for generating an action potential. Nonetheless, there is a depolarization, and this is called the excitatory postsynaptic potential. Now, what happens in the case of an inhibitory synapse? Depending upon the nature of the neurotransmitter, those inhibitory neurotransmitters tend to hyperpolarize the postsynaptic neuron. And how does it do that? These inhibitory neurotransmitters sits on these particular channels, which lead to the opening of the potassium channels, and hence the potassium channel ions move out, whereas the opening of the chloride channel leads to the moving in of the chloride ions. This leads to the shift of the resting membrane potential further towards the negative, that is, further down away from the threshold, and thereby this makes the postsynaptic neuron less susceptible to fire upon stimulation. And this is what is called the postsynaptic, inhibitory postsynaptic potential, or IPSP. Now, as we observed in the last case, a single EPSP, unless it crosses a threshold, cannot trigger an action potential in a postsynaptic neuron all by itself. So when does the EPSP lead to firing of a postsynaptic neuron? If there are multiple such axon terminal EPSPs which are reaching from multiple axon terminals which fire on the same postsynaptic membrane, and if there is a small number of terminal in which delivers these impulses very rapidly. So this is the principle of neural integration. So what the postsynaptic neuron does is it receives all these information from the presynaptic neuron, that is EPSPs and IPSPs, and depending upon their summation, it decides whether an action potential will be generated which will travel down its axon. So this is a case in which no summation takes place. Why? Because the slow firing of the postsynaptic neuron EPSPs are far apart in time, and therefore two time separated EPSPs do not summate, and hence the threshold is not reached and no action potential is observed. On the other hand, rapidly firing presynaptic neuron EPSPs, which are close in time, they do add up, and this leads to crossing of the threshold and the generation of an action potential, and an action potential is fired. Now, similar is the case which can happen with spatial summation. Now, imagine if there are multiple neurons which are presynaptic neurons which are making connection with this single postsynaptic neuron. So what happens is more than one presynaptic neuron fires simultaneous EPSPs at different location on the same postsynaptic neuron. So two EPSPs in the same time appeared from different locations. This leads to the summation of the EPSPs and an action potential is fired. However, what happens if there are two presynaptic neurons, one brings in a EPSP and other brings in an IPSP. That is, the presynaptic neuron creates IPSP overriding the EPSP from another neuron. That is, you have an excitatory postsynaptic potential which causes a depolarization, as well as an inhibitory postsynaptic potential which causes a hyperpolarization. This stronger hyperpolarization compared to the depolarization actually nullifies the effect of the depolarization, and the IPSP and EPSP actually cancel each other. So in order to summarize what we have discussed till now, the longer duration stimuli can lead to initiation of multiple actions. The pulse frequency modulation theory suggests that the frequency of action potential is dependent upon the intensity of the stimulus, and the nervous system therefore encodes different intensity of the stimulus in terms of changes in the frequency of the action potential. The depolarizing graded potential called excitatory postsynaptic potential occur at the postsynaptic membrane, whereas the hyperpolarizing changes in potential, which are called the inhibitory postsynaptic potential, they occur at the inhibitory postsynaptic membranes. And according to the principle of neural integration, the summation of the EPSPs and IPSPs which reach a particular postsynaptic neuron determines whether that neuron will be stimulated and an action potential will be generated and will be passed on down the neuronal tract.